Hello and welcome to this week's What Were They Thinking? Among the news items we have for you this week are the effects of COVID-19 on fertility, specifically looking at males, the unintended and somewhat unexpected impact of vaccinations on other coronaviruses other than those of COVID-19, possible benefits to allergy testing for children and possible ways to make them less likely to develop those self-same allergies, the surprising frequency of death after sex. These and other news items can be found at the timestamps given below. Let's start with fertility and COVID-19. Last week, we spent some time talking about the positive benefit of being vaccinated if you are pregnant. That is, unvaccinated individuals last week had a 4 times higher chance of miscarriage or similar deleterious birth event than those who were vaccinated. Unfortunately, the problems don't just end there for those who get infected with COVID-19. If you're a male, you are going to have lower fertility. We've already mentioned how men are going to have complications in their nether regions with things like blood clots in testicles, thromboses in veins, and so on. All of these will affect your sex life and ability to reproduce. The research broadly found that if you are a male and you have an infection with COVID-19, you had a roughly 18% lower chance of achieving pregnancy. Although this figure is small, it is statistically significant and will be all the more significant for those who are trying to get pregnant. This same correlation was not found with women though. Females infected with COVID-19 do not appear to have a lower likelihood of conception. We've already spoken about the issues surrounding longevity and effectiveness, or at least probability, of pregnancy carrying through for women. For men, it's simply the ability to have a pregnancy in the first place. More than just the positive benefits for reproduction, it appears being vaccinated for COVID-19 carries with it benefits for the seasonal cold viruses. These are your common fever and headache-inducing varieties. Not necessarily influenza, but the common cold virus. These are a very wide family with many different types, and they mutate readily. This is one of the big reasons why they have always been hard to create a vaccine for, and why we've by no means ever been able to get close to eradicating it. The small study looked at 24 individuals who were given vaccines and their blood before and after. What they found was that if they were given a booster vaccine, they had a much higher antiserum generation to the common cold virus than those who didn't. This would indicate that if you have immunity to COVID-19, you're likely to have some degree of protection against the common cold. Going from COVID-19 related immunology news to autoimmune disease news, particularly around allergies to peanuts. Allergies to peanuts amongst children are an increasing thing. This obviously leads to a variety of issues, especially if the children are extremely allergic, so much so they go into anaphylaxis. This is often why most schools have now begun banning things like peanut butter sandwiches, why various things like EpiPens are required to be on site. There's a whole bunch of modifications that need to be made to the environment to protect these children. The problem is we don't know why the allergies are increasing. What we do have is a variety of attempts to stop the allergies once they've been identified. This study was just such an example. It took nearly 150 children, it gave 2/3 of them a peanut flour based product that was mixed into food so it wasn't identifiable. The 2/3 of children who did receive it appear to have had a large percentage that were no longer allergic to peanuts at the end of the trial period. That was about 2.5 years of treatment. This means that they were able to train the human body not to recognize the peanut proteins as something foreign and begin attacking it as an allergenic substance. Although 70% of the group that was originally tested was able to get into remission and stop being allergic, unfortunately only 21% stayed in that category 6 months after they'd stopped treatment. This is still an incredibly high rate considering you're trying to train something in the human body that's not meant to be able to be modified that drastically once it recognizes something as foreign. If it could, bacteria and viruses would have a much easier time of things. While still on the topic of food allergies, but shifting slightly to the side, this time looking at food sensitivity tests. Food sensitivity is a somewhat nebulous term. You realistically only have food allergies or not allergies. Sensitivity is something that's often used for marketing and isn't well established. There are things like low FODMAP foods that have low certain proteins within them that aren't gluten and so are distinct from celiac disease, but cause some sort of gastrointestinal issues. These are still a kind of allergy. But even so, that represents a large part of the group who thinks they are gluten sensitive. Unfortunately, there are people who will jump on the idea that you can have sensitivity to food and begin selling products that are meant to identify this for you. Often these are at-home food sensitivity tests, and the question is whether or not you should buy them. The article from today looks at this, and while looking at it, it explores whether or not there's actually any value in paying in excess of $100 in some cases, or as little as $69 in others. Unfortunately, the tests that were examined in this article often follow the same sort of predatory practices as many others we've seen in the past, relying on a quasi-scientific approach that is often bogus. In this case, one is the generation of antibodies in blood. The second is resonance within food. As we've previously identified, these are nearly always going to come up bad because the science isn't there to support it, and there's no mechanism that can explain how they reach the standards that they reach. For example, for an antibody response to occur, you must have ingested the food and had the immune system identify it and begin responding by producing antibodies. The resonance one is just pseudoscientific nonsense full of catchwords. For further examples of just how this sort of research is somewhat faulty, but also how it can be done right, we've linked a video in the top right hand corner on gluten sensitivity and one of the researchers that identified it and then went back to figure out why they were getting the results they were. Next is an interesting article from The Conversation, and it's from a pathologist explaining why there's an increased risk of death after having sex. Generally speaking, sex is something that is quite messy and violent within the human body. It increases the heart rate, muscles spasm, the nervous system is activated in all kinds of ways, hormones rage, neuronal signaling goes wild, and more. This obviously is going to be very hard on the body. And in fact, so much so that dying during or shortly after sex accounts for just over 0.5% of all cases of sudden death. Obviously, you can't just look at this and go, well, these people have died, therefore it's related. You need to begin looking at not only long-term data, but very specific data, and that's what a German study has done. They looked primarily at men aged around 59 years, and the cause of death is a heart attack. They then looked at other studies in other countries and found that these men either had just or were in the middle of sex. Not just men experience this, and that's another consideration. Another study found that 35% of the cases they looked at were in women. That is a much higher figure. Once you begin exploring the various causes as they're given in this article, begin to see that there's a lot going on. To use a quote, "It's violent, it's ugly, and it's messy, and if God hadn't made it unbelievably fun, the human race would have died out eons ago." Unfortunately, the quote also happens to miss the fact that quite a few men and women are dying because of it. Going to medical news that is far more relevant and important. It's the treatment of a 700-day long infection that has been resistant to antibiotics. This was only made possible through development in an area of research called phage therapy. Phages are what you would consider viruses that specialize in infecting bacteria. By being able to select pre-adapted or specially developed bacteria targeting viruses, they are able to more effectively attack only the bacteria and leave the host body unaffected. This means you can begin destroying more of the bacteria quickly and help to support the immune system as it's trying to take out the infection. That's why they were able to take care of this 700-day long infection for somebody who had had a treatment or antibiotic resistant infection, something that's often incredibly hard to take care of. This particular study differed from most other clinical trials that have tried something similar, in that it had a customized phage therapy designed behind it. That was, they selected only those that were developing in the site of infection and specific to the strain of bacteria that had infected the host. Most other research has relied on a broader cocktail of different phage types that are able to target the bacterium but may not be especially suited to it. Where viruses designed to specifically target bacteria are one thing, creating something that size from scratch is another. That is just what researchers across the world have been trying to do for some time now. Create tiny artificial organisms. Obviously, being such a difficult area to work in, you have to start with the most basic of things that you can. Whether that is simply proteins that self-assemble, we've managed that much. Whether or not it's something more advanced, such as being able to get things to move in the right way to chemical signals, something we've also managed. But a whole organism, that is all working parts together, that's a different matter entirely. Having had these relatively more fundamental processes well understood, there have been several attempts at creating a fully synthetic organism, with every unneeded gene stripped out and only those that are absolutely essential for life remaining. This means the organism itself can't seek for its own food. It only has to replicate and grow. Other than this, it's very hard to get accurate and reliable synthetic organisms. That's why rather than continuing to run at these very difficult to develop synthetic organisms, some people are trying to model it and then build from that kinetic model and figure out how to design the most minimal cell from that. Where genetic modification by paring away every unwanted gene is one possible option. The opposite is genetic modification for crops to make them more resilient, more productive, and so on. These genetically modified organisms are often seen as somewhat negative by many people. Often this is due to fear of the unknown, and as a result many countries have often implemented rules that have severely limited not only the number but nature of genetically modified crops that can be grown and sold. In the UK, these rules might be pared back now, hopefully with the intent to allow for the cultivation of genetically modified crops in the fields, or at least field trials. There is a simple reality. More food is going to be needed as the population of the world grows, especially as the population of the world grows and is concentrated in areas where there is less farmland available due to population density. This means unless we are going to be moving food halfway across the world again, it's unlikely to be sustainable without much better crops available. Things like the ability to have more weather-resistant or drought-resistant food is important in that respect, and the only way we're going to get that soon enough to be able to provide for all of humanity as we can at the minute is through genetic modification. This is where the UK's decision to increasingly allow the movement towards gene-edited crops and field trials is going to be incredibly important. One of the reasons for this is likely that having now left the European Union, the UK is able to make much more free decisions, especially around genetically modified crops, where the European Union has a lot of rules about them. And this could make it possible for the UK to be much more independent when it comes to food from the European Union than they were before Brexit. Our next piece of news moves out of the genetic modification and biology fields to physics. It's also a record-setting piece of news. A single bubble has been kept intact for 465 days. You may not find this very exciting after all. Many people will look at something like a soap bubble and know that yes, it will eventually break after a few hours, maybe a day or thereabouts. The thing about soap bubbles is that they are a very interesting chemical structure, and the way they keep their shape and eventually lose it all owes to how the different parts of the chemical called glycerol that make up the soap bubble work. The various tension within that bubble is what keeps it in its shape. As that tension is gradually released, the bubble breaks. The longer you can keep the bubble intact, theoretically the better, depending on what you're going to be using it for. That's where this research comes in. It used microparticles that had inhibited the drainage of water out of it via gravity. They also added more glycerol that gave it a more steady state. By doing this, the water would not evaporate out, and it would instead hold onto the water. This would allow the water to stay within the bubble and keep it tense. That tension would then compete with the environment, or the ambient air, and it would stay as a bubble. The final news item we have for you this week is that about SpaceX's abandoned rocket, a rocket that's about to crash into the moon. In 2015, NASA contracted out to SpaceX the deployment of its Deep Space Climate Observatory. This satellite deployment was relatively standard. Send a rocket up into space with the satellite in its end, put out the satellite in the right location, and then call it a day. Unfortunately, this process also creates waste in the form of the second stage of the rocket. This more or less sits in orbit, just floating about doing relatively little. The rocket did, however, have a certain amount of inertia still, and since there's very little pushing back against it in space, it was going to continue traveling on its current trajectory. Based on calculations of its trajectory after the moon modified its orbit just slightly, it's going to crash into the moon's far side somewhere around March 4th. The problem is that this is not an exceptional event. In fact, there are many pieces of waste still left up in space from the earliest days of space launches. We of course also have discontinued satellites. There are things like the waste left over after the satellites that China destroyed recently with their surface-to-space satellite-destroying missiles. There is various other damage, debris, and so on. All of this is adding up over time, and that's going to be very important in the near future with the increasing number of satellites and other material and equipment sent into space. That's all the news we have for you this week. Thank you for watching. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.